So, an extension of somatic embryogenesis, we will be talking about synthetic seed technology. Now, synthetic seed technology involves somatic embryogenesis for which is needed for large scale propagation of plant material. Now, even somatic embryos can be used for clonal propagation of plants, and they can be genetically stable depending on whether they are indirectly produced or directly produced from the explant. Now, large scale propagation requires what? Planting material of suitable age and of high germination rate. So, a large number of somatic, the advantage of somatic embryogenesis is that with the same genetic composition you can produce a large number of plantlets in vitro by scaling up somatic embryos. So, either you can have embryogenic cell lines and then you scale up the embryogenic cell culture and then in a two-stage cultivation convert it into a somatic embryo form. So, the technology is now in synthetic seeds, what is, you cannot directly use in vitro conditions, you can directly use the somatic embryos and regenerate into plantlets. But if you if you want to use it outside as seeds for planting material, then generally it is encapsulated. Now, encapsulated, there are 4 ways in which synthetic seeds are produced. One is uncoated desiccated somatic embryos. This is one way of producing synthetic seeds. Then coated desiccated somatic embryos. Now, desiccated means what? Dried. Then encapsulated hydrated somatic embryos, which means the encapsulation is a hydrogel or which can provide water content. Then uncoated hydrated embryos. Now, there are advantages, merits and demerits associated with all these 4 types depending on the dehydration tolerance of the species of the somatic embryo. It can be decided whether to encapsulate it, not encapsulate it. So, you need to make sure whether the encapsulation, there will be a balance out between its vigorousness to convert the time it takes to regenerate into plantlets and the protection from the environment such that it does not become non-viable. So, let us talk about the coatings. Uncoated desiccated somatic embryos, what are they? Somatic embryos, they are dried to reduce the moisture content up to 8 to 15%. So, the moisture content is reduced such that it is observed that they can remain viable up to a period of 12 months, which means they can be stored for a year long period. Now, somatic embryos, they are, in comparison to seeds, they are less vigorous. So, vigorousness means what? The capacity of survival or remaining healthy in order to regenerate into a new plantlet. Now, the survival rates of the embryos, it also depends upon the desiccation tolerance, which means how much that species is tolerant towards dehydration or loss of water. Now, in that case, if suppose a species or the somatic embryo is sensitive, then hydrated coatings are used. Hydrated coatings, they are generally, for example, made of hydrogels. What are hydrogels? For example, very commonly used when you make your beads. What kind of beads do you make? You use sodium alginate, mix it with calcium chloride to make calcium alginate, and which is a gel bead. So, hydrogel means what? Hydrophilic. So, which means a lot of water, they can swell because of the absorption of water. Now, this water is nothing but a dispersed phase in the cross-linking, that cross-link matrix of the polymer and the cation which you are using. So, desiccated embryos, they have, please remember, they have poor conversion, while hydrated encapsulated somatic embryos, they have higher conversion frequencies, sometimes similar to that of the uncoated somatic embryos. So, what all can be used to generate more number of somatic embryos or to increase the vigorousness of the somatic embryos? People have used amino acids, organic acids like aspartic acid, malic acid, so in the form of salts, potassium malate or potassium aspartate, these are called as, even succinate. So, these are called as organic acids. So, supplementing the media with these organic acids has also helped in increasing the number of embryo generation and even the conversion of embryos. Now, abscisic acid, which is ABA, abscisic acid has been found to improve the development of embryos. There are 4 different stages we learnt in somatic embryos. So, the rapidness through which a somatic embryo converts it into a cotyledonary stage, where root and shoot primordium is formed, and which leads to regeneration of plantlets. Now, what all other factors which can affect is oxygen concentration. Because if the seed becomes encapsulated, there is high chance that there will be mass transfer limitations, and which factor becomes mass transfer limited is oxygen, because oxygen is sparingly soluble. So, steep oxygen gradients have also have been found to be limiting the somatic embryo regeneration. So, in that case, as you use in fermentation technology, oxygen vectors are used. These are nothing but organic compounds like your, for example, you will hear dodecane, or these are nothing but hydrocarbons or hydrofluorocarbons. So, why are they used as oxygen vectors? Even in fermentations, you will find that oxygen vectors are added in the medium to improve the oxygen transfer capacity. Now, how does it improve? Because these have higher solubility of oxygen in comparison to water. Like in water, how much at ambient temperature and pressure? 8 mg per liter around. So, but for example, in dodecane, it is around 55 mg per liter. Then in hydrofluorocarbons, it can go even up to more than 100 mg per liter. So, depending on the size of these hydrofluorocarbons or hydrocarbons, you disperse them into the aqueous solution, they will form a film around the gas bubble. So, what all different phases are now involved? The gas bubble to this oxygen vector film, from the oxygen vector film to the aqueous layer, from the aqueous layer to the cell. So, now because what will improve? The driving force improves, isn't it? Because there is higher oxygen transferred from the gas to the oxygen vector film. So, thereby leading to better oxygen transfer rates. So, these are also sometimes preferred, but depending on what is the droplet size, how it is dispersed in the medium, that is also a critical thing. So, agros, what other these hydrogels are used, materials, agros, sodium alginate, polyoxyethylene. So, they form a film which has a non-damaging effect on the embryo. So, hydrogels, I have told you what are hydrogels, water loving polymeric matrix, so which absorb water. So, water is the dispersed phase in these matrix, polymeric matrix, which then swells up. Now, let us talk about germ plasm conservation. Somatic embryogenesis or synthetic seeds can also be used for germ plasm conservation. Generally, seeds are used for germ plasm conservation, but there are demerits associated with it. We will talk about it. So, first thing, what are the different ways in germ plasm conservation is done? One is called as in situ conservation, the other is ex situ conservation. Now, in situ preservation of the plant material is using your biological national parks or reserves, biosphere reserves you will find. Then gene sanctuaries are where the original plant material in its natural habitat is maintained. Now, there are limitations to this kind of conservation process. The limitations, logically what is it? One is space, but more than that, if you are talking about conservation, what will be the impacting factor on this strategy? Any geographical or environmental calamity, so you cannot save it. Then ex situ preservation is what? That you provide suitable conditions in the gene bank to preserve this material. For example, you preserve them either in the form of seeds, synthetic seeds, or different forms of in vitro cultures like even shoots, meristems or somatic embryos. But then conditions have to be provided, which means the media, how do you cryopreserve it or how do you preserve it matters, so that the viability is not lost during the prolonged storage period. We will talk about that. So, I said there is a demerit associated if you are conserving the genetic material in the form of seeds. The demerits are there, it can lead to seed dormancy, which is, for example, we were working with Viola odorata, that Banafsha plant, if you remember, for cyclotides production I was talking about, and we found it very hard. We got the seeds from one batch we got from UK and the other we were trying to procure from Australia, but we did our level best, but those seeds could not regenerate into plants. And the seeds, and it was known that Viola suffers from this demerit. The seed, generally the propagation of Viola odorata is not preferred through seeds because of the seed dormancy problem. So it is generally propagated vegetatively through runners, which is a part of the stem, lower part of the stem. So that is a problem. Seed dormancy can be a problem, seed viability can be a problem, then seed destruction by endogenous pathogens can be a problem. So where you can lose the genetic material. Then they are also confined to now seed. If you are using a seed for conservation, then it is confined only to plants which propagate through seeds. Plants which do not propagate through seeds, which are vegetatively propagated, then there has to be a way for conservation of such plant material. So vegetatively propagating plants, they are generally conserved using in vitro conservation techniques, where I said you used different parts like shoots, meristems, somatic embryos kind of cultures for preservation. Now the advantage is that you can conserve, as she was saying, one of the demerits, large space is required, but if you are using in vitro cultures, in small amount of space you can conserve or store a large amount of material. Then it overcomes the problems of climatic dependency or environmental dependency. Then provides large amount of material for culturing, for regeneration later. So cryopreservation. Now once we are talking about conservation, for storage you need to store it under right conditions and properly, so that the material remains viable during the storage period. So for that, cryopreservation techniques are important. So how do we cryopreserve plant materials or any kind of cell cultures? Now cryopreservation is what? Preservation under sub-zero temperatures, freezing temperatures, sub-freezing I would say. So generally you will find, whether it is animal cells, plant cells, microbes, liquid nitrogen. So for plant cell cultures, animal cell cultures, the biological material is stored under liquid nitrogen or in the vapors of nitrogen. You can, why are we storing the material at such low temperatures? Because it has been observed that temperatures in the liquid nitrogen, which is so low, sub-zero, entire metabolic activity of the cell can be brought to standstill, can be arrested. So what is preferred? You gradually first reduce the metabolic activity and then bring it to standstill. Now then what is the right way? You take the biological material and put it in liquid nitrogen. So it can be stored, but there is a demerit. This is not how it is stored, especially the cells where viability is important. Now why do you think it is not stored as it is, bringing it in and storing it in liquid nitrogen? Cell viability is adversely affected under sub-zero temperatures. The first question which arises is why? Very nice. So there will be crystallization happening. So what will happen? It will form crystals, the crystals will grow, and in turn, as the crystallization of water might happen, many things happen. One is, there can be direct damage effects to the organelles. They can pierce the cell membrane, rupture the cell membranes or the organelle membranes, thereby reducing the viability. What else? There can be dehydration. What else? Dehydration in what different ways? Internal crystallization is more related to internal damage. So how does the freezing begins? Suddenly everything all together? So outside there is water getting frozen, then the cell. So what happens? There is a water, there is a water loss causing dehydration, and as you said, some effects due to the internal water also freezing, not being available. So then that also causes dehydration and loss in viability. So what is done? Cryoprotection is done before cryopreservation. So what is cryoprotection? We use cryoprotectants, agents which can reduce the damage because of cryopreservation. Now what are cryoprotectants? Cryoprotectants, now in nature also plants must be, because plants cannot move, isn't it? So extreme in cold countries, how do plants manage? That's called as, very good, that process is called as cold acclimation. So what does it mean? It means that entire genetic program is switched on, which leads to upregulation, downregulation of many proteins, and also which in turn leads to change in the metabolism, change in the growth, reducing the growth, balancing out the energy levels, the amount of PR proteins, which means especially antioxidants, then the cell membrane composition in terms of unsaturation of your lipid bilayer changes, then PR proteins, anti-freeze proteins are produced, sugars, alcohols, polyols, then proteins which can act as, or substances which can act as osmoticums, the cell starts producing. So similar thing we try to imbibe in vitro. So we add cryoprotectants which include glycerol, then you must have heard about the glycerol stalks. Why do we prepare glycerol stalks? That's the very reason. The glycerol stalk composition also varies. Sometimes you will see in literature people use 45% glycerol stalk, some people use 20% glycerol stalk, depending on what species you are working, whether you want to cryopreserve bacterial cells, whether you want to cryopreserve fungal cultures, whether you want to cryopreserve plant cells, the glycerol percentage keeps varying. So these are, or sometimes DMSO. Now these are called as cryoprotectants depending on the different nature in which they are used. Some are cell penetrating agents, some are cell dispersing agents which can easily get mixed with water and balance out the osmotic pressure. So what is cryoprotection? So I will repeat, cryoprotection is necessary for cryopreservation of the plant material unless they are naturally dehydrated. So generally when we say cold acclimation, which plants use, it is nothing but a slow dehydration process. You will find that suddenly the spring comes and you will find new buds coming in, new leaves starts coming in. So how is it happening? Because these auxiliary buds or these buds which will regenerate into new plant material are cryopreserved or are cold acclimatized. So through this process which I was talking about. So chemicals such as DMSO, which we use chemicals in vitro conditions, glycerols, various sugars, sugar alcohols, they help by protecting the living cells against the damage under sub-freezing conditions. Now the question arises, how? So how do they protect? These compounds, they do what? You increase, you must have heard during your 10-12 standard, lowering of freezing temperatures. How does it happen? You increase the solute concentration, the freezing point reduces of the solution or the solvent. So the same thing happens here. Now you lower the freezing point of water by adding these certain agents like sugars. So it lowers the temperature at which the freezing would first occur and can alter the crystal structure of ice even. So the colligative properties of the cryoprotectants, it minimizes the harmful effect by the crystallization of ice. So what all properties of cryoprotectants are essential? They have high solubility in aqueous phase, they should have, then they should be highly penetrating so that they can reach inside the cell, then they should be low toxic, should not be very toxic to the cell, So, the toxicity has to be taken into account, which means the amount of cryoprotectant you are using, the concentration, it matters, has to be optimized. So, what are the different ways through which these cryoprotectants work? The phenomena which can cause damage to the cells during cryopreservation. So, what is the phenomena? Mainly occurs during the freezing stage and includes solution effects, extracellular ice formation, dehydration and intracellular ice formation. So, what do we mean by all these four things? What is solution effect? An ice crystal which grows in freezing water, solutes are exuded, causing them to become concentrated in the liquid water. Once, as you people said, the ice is forming, so rest of the cytoplasm is getting concentrated. So, this high concentration of some of these solutes can be very damaging. Now, extracellular ice formation, how is it happening? When tissues are cooled slowly, water migrates out of the cell. We know because of the osmosis. Now, as the water migrates, it will cause dehydration. Now, when tissues are cooled slowly, water migrates out of the cell and the ice forms in the extracellular space. So, sometimes the ice which is forming in the extracellular space will also cause damage by pressing the cell wall and the cell membranes. So, generally you will find that when extracellular ice formation starts taking place, the intercellular spaces, they get widened. There is cell to cell contact. So, now you will find there is more gap and the water there gets crystallized and the cells are then, the cell walls and the cell membranes may get damaged because of the crystal formation outside. Now, dehydration, now we understand how dehydration would occur. Migration of the water causing extracellular ice formation can also cause cellular damage because inside now things are more concentrated. So, everything is interrelated, even the solution effects, the extracellular, everything is happening simultaneously. So, then intracellular ice formation, if suppose intracellular ice formation happens, then the crystal is growing, it can directly cause damage to the intracellular organelles. So, if I ask you in what different ways the cell damage or sub-zero freezing or cryopreservation may damage the viability of the cell, you should be able to tell. So, what is the procedure? The different cryoprotectants which are used are, one is called as permeating cryoprotectants, for example, DMSO, methanol, glycerol, they are highly soluble in water. Then non-permeating can be sugars, sugar alcohol, high polymeric substances, molecular weight polymers. Now, you can use, the best strategy is to use a combination of these cryoprotectants because the mode of action is different. Some are cell permeating, some can cause reduce the dehydration process by reducing the osmotic pressure, so can have osmotic effect. So, better the proposed solution is you use a mixture of these cryoprotectants. So, what is the general protocol for freezing? It is not, when you are freezing, it is recommended to do slow freezing, not sudden. It is not recommended to bring the biological material, dip it in your cryoprotectant and suddenly put it in minus 176. So, that adaptation is what? When you are doing a slow freezing versus very fast freezing. If I give you a hint, then when you do slow cooling, it will give the cell the time to equilibrate. This equilibrium, vapor pressure equilibrium, will in turn facilitate the internal water not to freeze but remain at subcool temperatures. The idea is to prevent internal crystallization. So, how does it happen? Once you lower the temperature, the concentration of the extracellular liquid increases as more water is converted to ice. Now, since the vapor pressure of the frozen solution is lower than the intracellular supercooled liquid, the vapor pressure of the slowly cooled cells reaches equilibrium with external ice by effluxing water. So, you are giving time for the cells, what you were saying, to adapt. So, thus slow freezing can prevent the intracellular ice formation and consequently the freezing injury by the crystal formation and then subsequently the crystal growing inside the cell. So, in order to avoid this crystallization, recrystallization, so we try to thaw it, which means what are we doing? To improve the heat transfer rate, there has to be a bigger driving force. So, you will be suggested to put it in a water bath 35 to 40 degrees C, or you thaw using your thumb from sub-zero, which is minus 76 to what is the palm temperature approximately? So, that is what is recommended to prevent this recrystallization of ice. So, generally for plant cells, what do we at a constant rate of 0.5 to 2 degrees C, we are at this rate it is slowly brought down to some intermediate temperature before it is immersed in the liquid nitrogen, and then rapid thawing is recommended. Okay. So, the applications of this preservation or conservation is you can have clonal propagation or same genetic material propagated later, and you can conserve the genetic material. Preservation of rare genomes can be done using this method, and free storage of cell cultures or even cell lines for even secondary metabolite production or any other commercial application can be done.